Alright, in this unit we're going to be focusing on basic cell information, looking at cell structure and function, talking about the type of equipment we can use to look at cells, getting into the history, who the scientists were that found out this stuff, what were they doing, that type of thing. You can find this information also in Chapter 3 of our book, roughly around page 70, 69, 70 through 91. So let's go ahead and get started by looking at this. Let's first focus on how we do measurements with these microscopes, and we'll start looking at the types of microscopes. So we use metric system in science. You should be good with that by now. You should have been told that in years past. If you haven't, we use metric system in science. Centimeter, hectometer, nanometer, those types of things, what we use. For science or for cell purposes, we're probably going to use micrometers, nanometers, those types of things, because we're getting into these very small details of a living organism. Alright, so we know we use microscopes to study these small things. These microscopes that we use are going to give us a picture of the structure and hopefully even function of what some of these small, either single-celled organisms or... Alright, if you look at the picture, you'll be able to notice just a kind of a scale. Alright, anything that's smaller than roughly 1 millimeter in size, almost down to where we're getting to 1/100th of a millimeter, or we're looking at micrometers. Okay, we can't see it without using some type of equipment. So this kind of lets you see a sequoia tree maybe up in the Sequoia National Park compared to a paramecium, which we really down past, like I said, down past 1 millimeter, we really can't see this stuff. So we have to use some type of device, a microscope, to see these single-celled, whether it be parameciums or other protists or bacteria, even smaller than bacteria, getting into viruses, what different viruses look like, down even smaller into protein structure, or down even smaller to atom structure. So we can use the different types of microscopes to see the different sizes of this stuff that we normally wouldn't be able to see otherwise. Alright, so our microscopes are usually characterized by their magnification and their resolution. Magnification just means how much bigger we can make that object. Okay, if you're looking at cell structure, for example. Our particular microscopes that we are going to use uses these types of magnifications. It has the very smallest it has three types: the small magnification, medium, and then large magnification. This is going to be 4 times, 10 times, and 40 times the original size. The eyepiece is always 10 times the magnification. So you can see if you add those two together, the small magnification is going to be at 40 times the original size, or 100 times the original size, or 400 times, depending on what power you use, what magnification power. The resolution is kind of like how crystal clear is your picture. How much HD are you running? Are you running 720p? Are you running 1080i? Are you running 1080p full digital? That's what resolution is going to show you. Just how much difference there is between two little small specks within a structure. Now we can increase resolution by increasing the magnification. Alright, so let's start looking at these. Our light microscope is the one we use in class. It's called a light microscope. It uses a light source. It shines it through a structure called a diaphragm or a base, so you can see light coming through that particular, shining through a particular cell or structure that we're looking at underneath the microscope. We call it, the version that we use is going to be the compound light microscope. And usually when we're looking at things, we're not seeing, the stuff isn't in this cool color type thing. Now, if it's a plant-based organism, then yes, you're going to see some green in the color. Or if it's a type of dinoflagellate that might produce some type of chemical color. There are organisms that do that, but in essence, we don't see, like some of these pictures that you're looking at, we're not going to see color like this. It has the things that we look at in our microscope have to be colored. They have to be dyed so we can see the different parts of it. That's why you see color. unless it's an organism that uses like chloroplast or uses some type of chemical process to produce a light, we won't see color. You'll see things like this picture where it's all the same color, but then we will dye it to get different structures within what we're looking at. All right, you step up and get a little bit fancy, a little bit more high tech, a little bit more money, and you get these electron microscopes. Okay, they have extremely high magnification, so they can increase the size of something that's a nanometer almost, big enough to where you can see it. Something that's actually going to be smaller than a cell. Something that's going to be smaller than a cell is in your body, or smaller than, say, paramecium, which is a protist. Something that's smaller than those. These electron microscopes will make them big enough to where you can see them. And they're pretty high-tech machines. Okay, this actual picture here is a picture of mitochondria. And we'll talk about mitochondria a little bit more when we go on in this unit. But mitochondria are little bitty organelles, little bitty structures inside a cell. So they're so small they can fit, hundreds of these can fit inside a cell. That's how small they are. There's no way we would be able to see that using one of our compound light microscopes, but an electron microscope has the technology and the power to make us be able to see that. A scanning electron microscope, and even fancier and even more expensive, even higher technology, it's going to give us a 3D image, a computer-generated 3D image of what we're looking at. So if it's a red blood cell, you're just going to see two-dimension under a regular regular microscope or a regular electron microscope. But a scanning electron microscope will actually give you a 3D image, let you see an actual picture of what that thing looks like in all dimensions. It's pretty cool how it works. Now, how do we find out about cells? Here we go. Let's start digging into some of this history. Back in the middle 1600s, right, a gentleman by the name of Robert Hooke first started describing cells because he used cork. Cork is a substance produced from trees. You use it. You can cork bottles with it. Wine corks, for example, is a type of cork that it's talking about. He used pieces of a cork and he looked at it under a microscope that he made, one of the original microscopes. Very basic, but it let him magnify what that piece of cork looked like, and he saw that there were small spaces and pores inside of it, and he called that cellulae. Cellulae just means rooms, is what it means, so, or compartments. So he was the first one that started looking and describing what cells were probably going to be based on his very first microscope. Van Leeuwenhoek came along a little bit later and took that information, and he, technology progressed a little bit more, and he actually was able to look at living cells for the very first time under a microscope. And then after him, Schwann came along and added on animal cells. So Schwann looked at animal cells, but Leeuwenhoek was the first one to look at living cells. So Leeuwenhoek looked at living cells, and later after that, Schwann came along and described animal cells. But Hooke was the very first one as far as talking about cells, coming up with how the name of them, how things have these small little compartments or cells that they're made of. All right, let's get into cell theory. There were some guys that contributed to cell theory, and let's throw all those guys up there. Okay, Schleiden, in the early 1800s, him and Schwann got together. Schleiden talked about plants and talked about that plants using the basis of what Hooke did and Van Leeuwenhoek did. He looked at cells and concluded that plants were made of cells. A little bit after that, I mentioned Schwann already. He came along and talked about animals and how animals are made up of cells. And then about 20 years after these two guys started using what Hooke and what Robert Hooke did and Van Leeuwenhoek did, Virchow came along and put his part into this process and did his research and found that all cells are derived or formed from pre-existing cells, which means, in essence, your skin cells, once they get worn out, make new skin cells. And so cells come from pre-existing cells. And this comes about to form our cell theory. Cell theory, basic parts. All living things are made of cells. All cells are basic units of structure and function for an organism, which means our skin cells, our liver cells, our heart cells are the basic units for the structure and function of our body. Same thing for plants. Their cells are the basic unit and for structure and function. And that new cells are formed from or made from old cells. So like I said, our skin cells, once they die off and get used, they're going to make new cells to take their place. All right, let's start looking at some common features. All cells have membranes. Okay, all cells have membranes. Plants and bacteria have something called a cell wall. Animals are just have the plasma membrane. So plants and animals both have a plasma membrane, but plants and bacteria also have a cell wall that surrounds their plasma membrane. It helps with protection and adds support, that type of thing. All cells have genetic material in them, which means they all contain a nucleus, and they all have, all cells contain genetic material. Not all of them have a nucleus, but they all contain genetic material, which is DNA and ribosomes. And then all cells have this cellular fluid in them to allow this stuff to be housed in, and that's cytoplasm. And then we're going to get to all of these parts as we go on. All right, so let's look at two major cell types. You've got prokaryotic cells and you have eukaryotic cells. Prokaryotic cells are single-celled organisms. They're a living organism. They function as a living organism, but they're only made of one cell. And we have to use microscopes to actually see these living organisms. Bacteria, for example, are single-celled prokaryotic organisms. We can't see them, but they grow everywhere. They're on tabletops. They're on your phones that you're playing with right now. They're underneath your fingernails. They're inside your mouth. They're on door handles, but we can't see them. And there's hundreds and hundreds of thousands of them everywhere, growing on top of each other, growing in these things called colonies. But they're still so small, we don't see them. Prokaryotic cells, they don't contain a nucleus. They don't have this main central brain, but they do have an area called a nucleoid, where the DNA is going to be comprised of, a central area, and then they have ribosomes. Remember ribosomes, we talked about this when we talked about proteins. Ribosomes are used to make proteins. Proteins, we've talked about functionality of proteins. Very important. So they have proteins, they have genetic material. They have a cell wall and a cell membrane for support, and they have some structures to help with movement. Where our eukaryotic cells are the most complex. All right, they have a cell membrane. If they're plant cells, they also have a cell wall. And then they have all of these smaller structures inside of them called organelles. The nucleus, they have organelles. All of these different things have different functionalities. But that's a eukaryotic cell. Plants and animals are eukaryotic. They're multicellular, and their cells have all of these little organelles in them to carry out specific functions. Whereas prokaryotic cells, they are single-celled, like a bacteria. They have some genetic material, they have some ribosomes, and that's it.